Hello and welcome back again to this channel, Callista's Bio Class. Remember that I told you I was going to come back with video on food test analysis for this year's Biology Practical 2025 Waec examination. Yes, remember that they asked that one of the specimens they required for Biology Practical is a raw egg. That should be specimen G, and then H, specimen H is water in a test tube. Now I told you that those two specimens are already driving towards food tests, and I'm here to give you give you a guide, like a standard on how you should answer food test question, how you should analyze your results, how you should be careful when conducting it, and then the words, the terminologies that should be used to write down your inference, rather start your test, write down your test, your observation, and your inference. Remember, it's an experiment, so you'll be analyzing it, or rather reporting it. And when you're writing a report on this, you should write it in a tabular form. The first column of your table should have the test, the second column should have the observation, and the third should have the inference, more like your conclusion. That's the meaning of inference. So I'm not going to waste much of your time, because I want this video to be short, so that while watching it you just get the idea what it's all about. So what I'm going to do is I'll be putting a picture and I'll be having a voiceover. Now that picture, it's where I've written out the analysis. So I'll present it, and in the background you hear my voice while I'm explaining. I'm here to talk about the Bureau test, help you, and also tell you why that water is there, because that water is going to serve as a negative word test. Don't worry, I'm going to do all of that in the video. So just stay tuned and you would enjoy this. Please don't miss out on this video, because food test is a must that it will come out in this year's 2025 Waec. Alright, so as you can see from the picture, the title, food test analysis for Waec 2025, that's for Biology Practical Waec 2025. So under the test, you see what I wrote. Most likely they'll ask you to crack specimen G, which is a raw egg. Now when they tell you to crack it, because we've already told your school to provide a petri dish, so I can crack it, of course, and put it inside, and a syringe. The syringe there is for something. It's for you to suck, or this, or let me use the right word, it's for you to collect liquids, just like in the hospital they use syringe, but there's always a needle attached to it. But in this case they won't put a needle attached, because if you're using a needle, it's just for you to pierce a vein. You need something tiny. Yeah, you're not piercing anybody's vein, but you're trying to take samples of that liquid. So as you're pulling in your syringe, drawing it, the plunge of your syringe, as you're pulling it back, you're collecting a liquid. Now in this case, they may ask you to use that syringe to collect the white part of it. That's the transparent part. That was how I wrote white part. I cancelled it. I wrote transparent part of specimen G. And that transparent part of specimen G, which is a raw egg, is called the egg white, or albumin. So that egg white, when you take it, they may tell you to take 2 ml of the transparent part of specimen G. They may tell you to take 3 ml, whatever quantity they tell you to take, make sure you take it. So when you take it, they will now ask you to you might ask you to dilute it with water, maybe a few drops of water. That means you use your syringe, collect water from your test tube, which is specimen H. Water in the test tube is specimen H. So when you collect water, if they ask you to put 1 drop or 2 drops, you add it to the egg white. Sometimes they may not ask you to dilute the egg white, because some of these tests, when it comes to using egg whites for Bureau tests for protein, sometimes you can dilute the egg white, sometimes you might not dilute it, you still get the same result. So back on, I'm still explaining the column for test. When you've done that, that plus sign means that when you've collected that egg white, you've collected it 2 or 3 ml, you pour it into another empty container. That one you've collected with your syringe, you empty it into another container, maybe a beaker or a petri dish or a test tube. When you empty it, for example, let's assume it's a test tube, because it's most likely it works better in a test tube. So when you empty it into the test tube, they will now ask you to add. That's the reason for that plus. That means you are adding something to that egg white that you've poured into a test tube. That egg white you collected with your syringe, you are adding something to it. That's the essence of that plus sign. So that plus 2 ml of NaOH means you're going to use your syringe again and collect 2 ml of NaOH, meaning NaOH is a chemical. What it means is sodium hydroxide. So you collect 2 ml of sodium hydroxide and add it into the egg whites that you've collected initially. Then they now wrote plus. They now tell you to also add. That also add means you're going to put plus. Don't write add. On that test, you write plus. Whatever you're adding together, keep adding plus. So plus 3 drops of copper 2 tetraoxysulfate 6 solution. So tetraoxysulfate 6 solution will be given to you by your school. Of course, they'll label it. So when they do, you take 3 drops. If they ask you 3 drops, you use it. They ask you 2 drops. Me, I'm just using 3 drops in this my video to explain. So you use your syringe again, or rather use your dropper for drops. There's something called dropper. You use it to suck liquids and then drop it in drops. But if you use your syringe, you're collecting it in mils. So when you've collected like few drops of copper, as I think they say few drops or 3 drops, you add it to that place where you have egg white plus what you've already added sodium hydroxide. You now add that what copper. Now something you need to know, when you add sodium hydroxide to egg white, it doesn't change color. And you know that egg is a gelatinous thing. It's kind of jelly like. It's not like water that is just doesn't stick together. Gelatinous tend to stick together. That's why egg is a bit dry or stretchy. So when you add, let me go back. When you add sodium hydroxide to egg white, there's no color change. But immediately you add that copper 2 tetraoxysulfate. Copper on its own is blue in color. That copper that liquid that is blue, light blue, is always copper. Your teacher will label it copper 2 tetraoxysulfate 6. So when you collect that copper, let's say it's 3 drops or few drops or 2 drops, and you add it to where you have egg white and sodium hydroxide, you will notice that immediately you add it and you shake it, the color will start changing from the color of copper, which is blue, to start changing to purple or violet. Once it does that, that tells you that there is protein in that egg white, and that thing is proteinous in nature. So under observation, you will now write violet/purple color was what formed. Then inference means your conclusion, what you concluded from this test. It means that protein is present. This is how you write it. Then I'm sure they will tell you to do another test using water, which is specimen H, water in a test tube. If they do, that's the essence of that II, the test under. That II tells you, they may ask you to take 3 ml of specimen H. Remember, they don't tell you take 3 ml of water. Do not write take 3 ml of water. Are you following? You should write take 3 ml of specimen H. That was why in the first one, you saw me where I wrote crack open specimen G. They may now tell you take 3 ml or 2 ml of the transparent part of specimen G that you've opened. Specimen G is egg white, raw egg. So they don't usually write the name of the specimen egg. They'll just tell you few drops of specimen G plus, and the only thing they write is the name of the chemical, like sodium hydroxide or copper. But GH, all those things, you will never see water as H. Neither will you see raw egg as G. You always see them using specimen G in place of the name, which is raw egg. So for the second test, they may ask you, that is why I told you that water is there to confuse you. The water is there so that when you test it, some person might write protein is present. It is wrong, because the color, when you do it, you will never get a purple or violet color. Are you following? So let me explain it. Now, if they ask you to take 3 ml of specimen H or 2 ml or any amount they ask you to take, you take with your syringe. Then they ask you to add sodium hydroxide. Then you write plus. If it's 2 ml of sodium hydroxide, you add. If it's 1 ml, you add. So depending on the quantity they ask you, you now write. For example, I'm using 2 ml, so it will be 2 ml of NaOH, which is sodium hydroxide, as you can see from that picture. So after that, they will now ask you to add also the third one, which is the copper 2 tetraoxysulfate solution. Depending on the amount, if it's 3 drops of copper 2 tetraoxysulfate solution, you add. If it's 1 drop, follow the amount that was given to you. When you add that, then under observation, you will notice that immediately, remember when you add sodium hydroxide, the color never changes, it's still transparent. But when you add copper 2 tetraoxysulfate, because copper on its own, that copper tetraoxysulfate is already blue. So when you add it, it will just remain blue. That's the color you're going to get. That's why I said under observation you get light blue color is formed. Are you following? Light blue color is formed is what you get. It will not be purple because it's water you're using. You're not using egg whites. There's no protein in water. Then under inference, that's your conclusion. You write protein is absent. Please do not write protein is present. You fail it. Then let's move to the next one where I have possible questions that I drafted or I generated from this food test. All right, so for the possible questions, as you can see from the picture, possible questions from food test, I on my own thought of questions that I know they can ask you from this food test, and you should have knowledge of it. The first one says, what is the name of the food test conducted in I above? By I above, I mean in Luffy, go back to that table I, whereby you use specimen G, egg whites. You not call the name egg white. Remember, I always call it specimen G plus whatever they ask you to add. So for that one where you use specimen G, the name of the test is what we call Buret test for protein. Please do not write Buret test. It's not correct. If you write like it's correct but it's not complete, if you write Buret test, they might not give you your full mark. So the actual name is Buret test for protein. Why is this so? Because that test called Buret test is actually to test for the presence of protein. Are you following? That is when you use it. Now, the second question they may ask is, what is the name of the food test conducted in I above? That is the second test in that table where we used water. Now, the answer is Buret test for protein. Now, remember, someone might ask, oh, this one we use water and you're still calling it Buret test. Remember, what gives the test its name is the reagent to use. Remember that water, you added what? After adding water, you added few drops or 2 drops, or any quantity. You added 2 or few drops of sodium hydroxide plus copper 2 tetraoxosulfate 6. So whenever you add sodium hydroxide and copper, that thing is always what the reagent they use for Buret test. Even though you're not going to use a protein source like egg whites, like in this case you use water, the fact still remains that the reagent you use is the reagent they use for Buret test. So therefore the name will be Buret test for protein. The only thing is that it tested negative because that source was not the source, or that specimen H was not what a protein source, rather it was ordinary water. Are you following me? But the test will still be Buret test for protein. Remember, it's not just Buret test, but write it in full. Buret test for protein. Then the third question I drafted is, what other tests can be conducted in place of the one carried out in I above? Remember that the one you carried out in I above is what? Buret test for protein. There are so many other tests. There are 2 other tests rather you can conduct to test if there is protein in a substance. And those other 2 tests are what they are asking you, that what other tests can be conducted in place of the one you conducted already. So the other 2 tests you can use to test for presence of protein are, the first one is what we call millions test or millions reagents test. The second one is xanthopriotic test. So both of them are what? Tests for protein. In xanthopriotic test, I think that's the test where they use what? The tetraoxosulfate acid. But in millions test, they use the what? Millions reagents. So that is another possible question they could ask you. There are still so many other questions they could ask you from this food test. But when I get them, I'll still bring up the videos for you. So with this, I believe I've been able to help you have a knowledge of how to answer your food test question in this year's biology practical Waec question for the year 2025. Please always stay tuned to this channel, and I urge you, whenever you follow my videos, understand 1 or 2 concepts, please do share with your friends. Please do subscribe. Watch the videos to the end so that I can count, or watch the videos halfway at least. And thank you for your support. Thank you for the amount of new subscribers I had. I really appreciate you coming to my channel, watching videos, and also subscribing. Thanks a lot. I love you guys, and you give me more reasons to keep making videos. If there are any confusions you have, you can drop them in the comment section. If there's any video you want me to make, probably you've read it and you don't understand the concept, you can also drop it in the comment section, and I will do well to create a video on that topic. I wish you success in your forthcoming exam. Stay tuned to Callista's bio class. Thank you. Bye.